The overall goal of this procedure is to efficiently isolate specific RNA with proteins that are associated with the RNA in vivo.
This method can help answer key questions in RNA metabolism and regulation, such as what is a distinct set of RBPs bound to a particular transcript.
The main advantage of this technique is the high efficiency of isolation, which allows follow-up identification of novel mRNA-associated proteins.
The RaPID methodology utilizes a yeast strain that co-expresses MS2-tagged mRNA and a fusion protein of an MS2 binding protein and a streptavidin binding protein. Grow 500 milliliters of yeast cells at 30 degrees Celsius in synthetic dextrose or SD selective medium to an OD 600 of 0.8 to 1.0. When the cells are ready, centrifuge at 3000 times G for 4 minutes at room temperature and discard the supernatant. Wash the cells in PBS and centrifuge again. Discard the supernatant and resuspend the cells in an equal volume of SD without methionine. Incubate for 45 to 60 minutes at 30 degrees Celsius to induce expression of the fusion protein. After 45 to 60 minutes, set aside 10 milliliters of the cells for RNA extraction and use the remaining cells for RaPID purification. To begin this procedure, add to the cells